Hello, everyone, and welcome to this AnaBios webinar presentation. My name is Chris Mathis. I'm the Chief Commercial Officer at AnaBios, and I'll be acting as the moderator for today's session. We have three speakers today. I will be introducing them, and it's my pleasure to introduce our first speaker is AnaBios CEO, Dr. Andre Getty. He will be providing an introduction, a presentation, and discussion on why AnaBios is interested in precision cut lung slices. Dr. Getty did his PhD in molecular and cellular physiology at the University of Verona in Italy. Did a postdoc at the Salk Institute. I first met him when he was the president of Aviva Bioscience, and was one of the three founders of AnaBios about 10 years ago, along with Drs. Paul Miller and Jack Reynolds. And so, Andre, take it away.
So, yeah, I don't want to take any time from our two speakers today. I just want to spend a couple of minutes to introduce what is AnaBios doing and why we're doing what we're doing. AnaBios is a specialty CRO and a leader in translational research. And what that means is that if you are in working in life science research, whether in basic academic research or drug discovery in industry, you're very likely facing what is known as the translational gap. That is the fact that most of the work is conducted in animal model, in various species. And so there's really a big question of how much of what is found in animal models is translatable to human. And so there are four key questions that needs to be addressed to fill the translational gap. The first one is cross-species physiological differences, which are often not well-defined or known. Pharmacology of the pathological state is also something very critical in drug discovery, which requires the establishment of animal models of diseases. And one big challenge there is that oftentimes we don't really understand the fundamental pathophysiology of the disease, and so the models that we tend to create in animals are either incomplete or absolutely misleading. The other issue that is often overlooked is the quantitative aspect of translation from animal studies to human. And when it comes especially to the potency of drugs across different species, this creates major problems when taking, for example, compounds into the clinic, because a drug may be particularly potent in animal, not so in humans. So there are risks of over or underdosing patients, which can lead to a lot of side effects and safety issues, or if one is underdosing, an apparent lack of efficacy. And the last aspect that is really challenging to address, relying exclusively on animal models, is the selection of the patient population. This actually goes back to what I said earlier about the pharmacology of the pathological state. The poor understanding of the disease mechanisms and the pharmacology of the drug in the disease conditions makes it very difficult to really understand what is the ideal patient population or subpopulation for any given drug. So the way we have decided to tackle these problems at AnaBios is by enabling the study of human organs and tissues at the preclinical stage. And so trying to generate as much data as possible that is really predictive of human clinical responses at the preclinical stage, using preparations that essentially recapitulate the physiology and pathology of human disease states. And this really allows for addressing the activity of a drug in the human target tissue, also understanding the activity in the context of pathological state, because what we are doing is essentially recovering human organs and human tissues from organ donors. And these can be either in a normal non-pathological state or come from donors that were affected by specific and known pathologies. And so this is what would allow us to study, for example, the activity of a drug intended for atrial fibrillation, specifically in cardiomyocytes isolated from a donor that was suffering from atrial fibrillation. We have the ability at AnaBios to recover and conduct studies on a wide variety of organs and tissues from the human bodies. And as I said, not only in the normal non-disease states, but also in the pathological state. And all of this is very important because provides a very accurate and predictive assessment of potency of drugs in human organs and human tissues. And obviously, this is all relevant not only to the assessment of potential efficacy of a compound, but also to safety. So specifically, the type of tissue and endpoints that can be measured in these preparations in vitro in the laboratory can address not only efficacy questions, but also safety questions. Therefore, by putting all of these data together, one can really understand what is the therapeutic window for a particular drug. Is there a specific population or subpopulation of patients within a particular pathology that could benefit from a given drug? And these data can be extremely valuable when designing the first-in-human studies, and obviously in identifying the ideal clinical candidate at the preclinical stage. So with this context in mind, the focus on translational research conducted in human cells and tissues, we started about a year ago this webinar series, of which today we have the first instance that is focused on lungs with two excellent speakers. But I invite everybody to stay tuned. We have also additional very interesting webinars coming up. And also, I would like to ask everyone to reach out to us and make your suggestions for the themes and subjects for speakers for future webinars. As you will see today, the quality of the presentations and the quality of the speakers is extremely high. And the goal is to really stimulate the thinking in the context of translational research, the utilization of oftentimes human tissue or cell preparations that can really advance the technology and strategy that are used for moving compounds from the preclinical stage to the clinics. Today on the lung, the focus is going to be on precision cut lung slices. This is a relatively novel technology, and the speakers today are really two leaders that have made major advances in this field. This is now a methodology that allows a variety of applications, from the study of asthma to drug toxicity, fibrosis, respiratory infections, which is something that obviously is very much on everybody's mind in these days, lung physiology more in general, and then lung immunity and COPD. So obviously, we're not going to cover all of these topics today. Some of it will be covered today, but also this will be a very interesting introduction to the technique itself, because as I said, the two speakers are our leader in this particular area. So without taking any further time from the presentation, I will return to Chris for the introductions to the speakers.
Thank you, Andre. And just a reminder to the audience, we will be taking questions. So at the end, so you can ask a question from Andre or any of the speakers today by just typing it in, and we will get to that towards the end. So it's my pleasure to introduce our next speaker, who is Dr. Rama Krishnan. And Dr. Krishnan, he earned his PhD in mechanical engineering from Columbia University, then worked as a biomedical engineer at Exponent Inc. in Philadelphia. So he also has industry experience. He returned to academia in the capacity of a postdoctoral fellow at Harvard School of Public Health. He was later promoted to a research associate and then to research scientist. 2011, he joined the Department of Emergency Medicine at Beth Israel Deaconess Medical Center and became a member of the CVBR. So as you can see from his slide there, the title of his presentation is PCLS, a window into airway function. Take it away, Rama.
Thank you, Chris, and thank you, Andre, for the introduction. And thank you all for attending this webinar today. What I want to do is I want to start with a picture of a slice of a human lung. This precision cut lung slice, these are approximately 200 microns thick, and they're living. Nearly all the cell types, the 40 cell types that are present in your lungs, are present in that PCLS. What you see there, that's the airway, that's the epithelium on the inside, and around the airway is the airway smooth muscle. So we're going to take that human lung slice, and we are going to run an experiment. And I want you to keep your eyes on the airway. I will first stimulate this lung slice with a contractant, and then we will breathe it. And you keep your eyes on that airway. So here we go. Contractant and breathing. So what I hope you noticed was that when I added the contractant, that airway became smaller, it constricted, but then as I was breathing it, it became bigger and bigger and bigger. And in fact, if I waited another 100 frames, that airway, constricted airway, would get back to its pre-constricted baseline. Now, don't take my word for it. Let me show you data collected over many lung slices from many different human lungs. On the y-axis here is the reversal of constriction, and on the x-axis here is the stretch that was imposed. Don't worry about the differences between these three lines, not important today. The message is, the more the stretch that's imposed, the greater the reversal of bronchoconstriction. For example, at this high stretch, which is a very deep breath, you almost get complete reversal of bronchoconstriction. Now, the data so far are from non-asthmatic human lungs. We have anecdotal data over one asthmatic human lung slice. And now, using this lung slice, I'm going to run that very same experiment, and keep your eyes on the airway. First contractant, narrows, and then breathing. That airway stays constricted. Breathing does not dilate the constricted asthmatic airway. And this is in fact one of the major issues in the asthmatics. When they have bronchoconstriction, deep breaths are simply not possible. The airways stay constricted. From these and many other studies that we've performed, we've now understood that a major end effector for this difference between normal and asthmatic is the airway smooth muscle that surrounds the airway. In fact, what we have learned is in response to a stretch that the airway smooth muscle fluidizes. And by fluidization, I'm referring to a physical response that is characterized by relaxation of the contractile forces of the airway smooth muscle. And when that ASM surrounding the airway relaxes, the constricted airway is able to open. And we've hypothesized, therefore, that in asthma, it is the failure of this airway smooth muscle that surrounds the airway. It's the failure of that muscle to fluidize that is the proximate cause of bronchoconstriction. Now, this is actually a pretty compelling hypothesis because it brings together studies done over, in fact, the past 150 years, really trying to understand the difference between the normal and the asthmatic lung. So the hypothesis is compelling, tools are emerging, and the PCLS appears primed for us to uncover this important mechanism. But first, we had to overcome a logistical problem. My lab, and typically most labs across the world, we get 3, 6, I guess at most, human lungs a year. From a given lung, you can obtain hundreds of slices, so that's wonderful, and each of those slices can be independently manipulated. But for a given experiment, such as these stretch experiments, we end up maybe using 10 slices, 15 slices at most, because the slices live for approximately 2 to 4 weeks at most. So we get these lung slices, we've been waiting months and months, we get a lung, we get hundreds of slices, but we end up using 15 and checking out these remaining 85 precious tissue slices. And this is heartbreaking, and it's been a problem for us and many other labs, is how do we enhance the utility of this method? To combat the issue, we have devised a method of cryopreservation. Now, to those in the audience who are used to doing cell culture studies or any familiarity, the method for PCLS, cryopreservation, is precisely the same. It's well established in any lab that does cell biology. You just start with your slices, you dunk it in a media with DMSO, and freeze these vials slowly, first -80 and then liquid nitrogen for storage, and you can store them for days, months, and actually even years. Day of the experiment, thaw out rapidly, and you're ready to run your experiment. Pretty straightforward methods described in this publication. While we were running these studies, we wanted to first confirm that we aren't affecting viability of our cells in the PCLSs. I'm showing you here now the top panel is 10x imaging, bottom panel is 20x imaging. Column on the left is bright field images of the PCLSs. First row is never frozen. This is frozen thawed tissues. These are in mice slices, by the way. This is staining for live, and this is fluorescent staining for dead. You can immediately see live staining is predominant, dead is hardly present. Overlay, it's just all live staining, be it 10x or be it 20x. So this at least visually confirmed to us that the freeze thaw is not affecting cell viability. We were more quantitative about this. We used other assays to measure what is the percentage of dead cells using molecular measurements in our tissues. And the bottom line is irrelevant, really. It's 1%, hardly, and it's similar between the two groups. So our method isn't killing any cells in the lung slices. Neither is it affecting morphological changes. So this first row there is never frozen, frozen thawed, staining for the club epithelial cells, staining for smooth muscle cells. It looked similar. We weren't affecting morphology. And the most important question for us when we were running these measurements is contractile behavior of the airways. And to answer this question, we mounted a more careful study. We collected 100 slices from 4 different mice. They were divided into 2 groups, never frozen or a frozen thawed group. And we took each PCLS from each group, we contracted it first and then relaxed it with known compounds. And in order for us to compare these data, we took all of these outcomes and we normalized on a PCLS basis. We normalized it to its own baseline value. So I'm showing you here now the average changes. On the y-axis is the normalized change of contraction to its baseline. And on the x-axis here is the contracted across increasing doses. One curve is for the never frozen group, the other one is for the frozen thawed group. So you look immediately, you will see with the bigger dose of contracted, there is more airway constriction. But importantly, these are not statistically different. And the same story holds for relaxation. They both relax similarly. We went a step further. We looked in particular at the very small airways, the distal airways, the small sized airways, and again, they look very similar. So from these and other studies, we confirmed that cryopreservation preserves the viability of the PCLS, preserves contractile function in the PCLS, and enabled by these, we now have banks of tissues. We have mouse, rat, pig, and even human, which we can just thaw on demand and do our experiments. So that was significant because we overcame the logistical problem of tissue numbers. The next issue we had to overcome was the measurements, technical. It was the measurements themselves. When I tell you things like the muscle fluidizes, it relaxes, ideally in our assay, we want to directly measure the muscle contractile force and directly measure fluidization. So we wondered, can we enhance or develop new methods where we can directly measure these mechanical behaviors? And to do that, we have recently developed a method called tissue traction microscopy. To run this method, we start with a substrate that is soft. And by soft, I mean a few kilopascal stiff. The non-engineers in a room, a kilopascal is like squeezing toothpaste between your fingers. So it's really that soft. But toothpaste, you squeeze, it flows away. Unlike that, this substrate, you squeeze, it deforms, but when you let go, it returns to where we started. So this substrate is elastic. The substrate is also transparent, so you can look through and you can look at pictures on the surface of the substrate, pictures of the PCLS that's on the surface. And in addition to all of these substrates are also tacky, they're adhesive. So you can take your PCLS and slap it onto the surface, so it's stuck onto the surface of the substrate. And the final thing we do is when we manufacture these substrates, we also embed fluorescent beads at the very surface of the substrate. So then during the experiment, you can look through the substrate and you can get pictures of the PCLS, and that's the PCLS, and that's the airway, that's the lumen, that's the muscle. So you can get pictures like that in the experiment, but you can also look at displacements of these fluorescent beads. For example, with acetylcholine, a contractant, based on the displacement maps of those beads and stiffness of the substrate, we could directly compute the contraction map of these muscles. So for that, this is the muscle in these hot spots are regions of biggest contractile force. And when we added the relaxant, you can see that those forces decrease and the lumen that had narrowed now becomes bigger in size. So you can measure simultaneously contractile force together with lumen area change. I'm showing you here now average changes measured over many different slices from different animals. These were done in pig PCLSs, porcine lung slices. This y-axis, force changes. So the black curve represents force changes, while the gray curve represents lumen area change. And this is over time. In response to the contractant, you can see that the lumen narrows. So the lumen areas, the areas are constricting, the lumen is narrowing, correspondingly the forces are going up. Conversely, when you add the relaxant, you can see the lumen enhances, so the lumen is expanding, and correspondingly the forces are going down. But when you look at these side by side, you can immediately see that the measurements of force is less variable. And that's really important because these assays, we're currently using them for bronchodilator drug discovery. So the fidelity of the endpoint is very, very important. And the fact that these are less variable is actually quite a technical advancement in terms of bronchodilator measurements. Next, we wanted to understand sensitivity. To do that, we used histamine contractant, and we measured it over increasing histamine doses. And similar to the last plot, here is change of force in black, change of area in gray. Focus in particular on the lower histamine doses. There is no change of area that's detectable. However, the change of force is significant. That is to say, our assay is also more sensitive to bronchoconstrictor stimuli. And finally, for any given PCLS at any given time, one can get maps like this. So in addition to getting an average force change, one can also measure local spatial temporal dynamic changes. So here's an example. So that's a PCLS, human PCLS. This is a force map at some given time. And I'm showing you here now the entire experiment, and I'm plotting the average force change. So that's force, methacholine, you can see forces went up. For formoterol, forces went down. Stretch, ASM fluidized, the airways became bigger, the forces went down, muscle contraction went down, and then over time it recovered to baseline. So this is an average change of force over the entire experiment. But we can do one better than that. We can look at individual points. For example, I was interested in seeing what happens to the hot spot of traction of this contraction force over time. And that's plotted in green. That's a time trace of that hot spot over the entire experiment. What happens to these cold spots in time? And that's the time trace in purple. So the advantage of these sorts of measurements is when you compare normal versus asthmatic, you can look at spatial distribution of these hot spots, spatial temporal changes of these hot spots, and ideally we can also superimpose these upon histology. What comprises these hot spots? What's different, normal and asthmatic? So we have unprecedented spatial temporal resolution of these changes. So then, to conclude, what I have described to you is a human tissue-based platform, so directly focused on human tissues, the precision-cut lung slice. It's also directly focused on physiological endpoints. In the measurements I showed you, I had focused on muscle contraction changes in the settings of asthma. And with emerging methods that we have developed, it's also well suited for bronchodilator drug screening and discovery studies. You heard about the methods and application in asthma, but this method is being broadly utilized by my lab and by others to other obstructive airway diseases, as well as fibrosis, cigarette smoke exposure studies. You hear about exciting applications for toxicity screening from Dr. Brain. It's also being used in allergy studies. Before I finish, though, it behooves on me to talk about COVID. As many of you may know, COVID science, current models of rat and mice are imprecise. Primate studies have ruled the day. You see them happening all over the world. But the problem is that these non-human primate studies are slow to perform, and they're also quite expensive to implement. And we also understand, given all the emerging science, that lung is the major site of entry for this COVID-19 virus. And the lung epithelial cells are the major site of infection for the virus. I postulate, given these considerations, I postulate that the human PCLS is ideally suited to bridge the gap between human cell-based experiments on the one hand and clinical trials, phase 3 trials on the other. And measurements, potential measurements with the PCLS could include measurements of viral infection using assays, examples of those assays, measurements of cytokine secretion, histological changes, analysis of molecular changes in these PCLS tissues. And at the very least, it can also be a safety platform for these emerging class of drugs, emerging class of vaccines. The PCLS can perform the important job of doing human-based safety studies, and human tissue-based safety studies. And important to note is in performing these measurements, you can do them at high throughput. You can run these in 96 well plates, for example, or whatever platforms that are typical to these assays, but you can miniaturize them to high throughput platforms. You can also multiplex them. So it's not as though one PCLS can be restricted to only one of these measurements. These can be run, many of these can be run simultaneously. And the measurements can be run either in PCLSs first exposed to COVID and then looking at treatment that overcomes existing infection, or conversely, you can have first expose it to a vaccine type drug, then post-exposed to COVID and see how well the vaccine is performed in preventing a viral infection. So you can do a whole spectrum of studies with these PCLSs. And with that, I would like to conclude. I want to acknowledge Nicole from my lab, my colleagues at the University of Chicago, Sumati, she ran those experiments in the human PCLS. She's currently at Pfizer. My colleagues at MGH, BU, Drs. Brain and Molina, and thank you all for your attention.
Thank you so much, Rama, for the excellent presentation. And just a reminder to those of you in the audience that if you have questions, you can write them on the lower right-hand corner of your screen. And it's my pleasure to introduce our next speaker, who is Dr. Joseph Brain. Dr. Brain received his medical degree from Harvard University. He's presently the Cecil K. and Philip Drinker Professor of Environmental Physiology at Harvard. His research emphasizes the body's response to inhaled gases, particulates, and pathogens. His studies extend from the deposition of inhaled particles to their clearance and health effects. Of particular interest is predicting the potential toxicity of new and complex materials such as metal oxide nanoparticles. So today, Dr. Brain's presentation will be on that subject. And the title of his presentation is, as you can see, Toxicity of Complex Formulations Using PCLS. Dr. Brain, the stage is yours.
Thank you. I'm delighted to be with all of you. And I think Rama has given you an excellent introduction to the technique and its many applications. And so he really stressed pharmacology, drug development. I found very interesting his ideas about how we deal with COVID-19 and connect the technology to that. I want to introduce you to another sort of universe of applications, and that's in relation to toxicology. And so I just want to give you an example of how we've used PCLS to look at the potential toxicity of various chemicals and also particles. I'm part of a Center for Nanotechnology and Nanotoxicology. And so that's one of our specialties, and that's one of the areas in which we found that PCLS is very, very useful. We were asked actually by the European Union and the National Science Foundation to look into nanoparticle containing consumer products. So here's an aerosol can. You might be able to buy something like this at your neighborhood hardware store. And this particular application was used for waterproofing your new shoes, your hunting jacket, spraying on the bottom of skis to make them more slippery. And it was thought that the presence of nanoparticles in this spray would have some real advantages. There were a couple of problems. Some consumers complained, particularly those who did this not just once a year, but did it 10 times a day. So we were asked to look into this. So this is sort of the context of the application that I want to mention. In this can is a mixture of stuff. So it's interesting that the nanoparticles that were of interest as to whether or not they were toxic or not is only 0.2% of what's in the can. And you can see there's water, there's ethanol, all these other materials were added for certain reasons to control the surface tension, to affect the particle size, the rate of evaporation, etc. So this was the problem that we were faced with. We were interested in nanoparticles in general, and you can see here in the studies that I will show you, we were actually able to look at 10 different nanoparticles of cerium oxide, silver, and then 3 other ones that are listed at the bottom here, 8, 9, and 10. And so these are the ones that we looked at. So again, this is daunting. If we relied on animal experiments, it would be expensive and would take a long time. But PCLS to the rescue was very, very useful. This is a summary of the methodology. And I think to a great extent, Rama has already sort of, in the introduction from the president of the company, has sort of described this. You prepare the PCLS with a vibrating microtome, and then you can either do experiments with the fresh slices after the slices have recovered overnight, or as Rama has described, an enormous advantage is being able to cryopreserve these. And in the data that I showed you, we both use them fresh, or we sometimes thaw them and use them. And we then looked at responses in the data that I will show you after 4, 24, or 2 days, 48 hours. And a variety of responses were measured: mitochondrial activity, lactic dehydrogenase, and a variety of cytokines. So this is the overall methodology that we used. So let me now mention a couple of questions that we asked and were able to answer with this methodology. These are the slices in a 24-well plate. We have also used, most of the data is actually from a 96-well plate. So again, what's exciting is that every single well is a unique experiment and can have different conditions, different particles, different concentrations. One can look at a variety of factors. And also there's fluid filled, and as I'll mention later, one of the things that you can do is to take the fluid from the slice after 1 or 2 or 3 days, however long the experiment is, and analyze it for what's, for example, for cytokines and chemokines that are in the fluid above the slice. So here's the first question. What's the relative potential of different chemicals to cause damage? And we have lots and lots of data. I've just sort of summarized them into 3 classes. So here's one class. These were relatively non-toxic, particularly if you look at the blue and yellow lines that correspond to PS and also to MEC, to TA. You can see that even at the highest concentrations, 12% in the media, they were really not toxic. The one shown in red, MEK, actually becomes a little toxic at very high levels. Now let me show you another class. Note that along the bottom is the percent of the chemical in the media, and it goes up to 12%. The vertical axis shows you mitochondrial activity in the cells in the lung slice, with 100% being the control. And you can see how relatively non-toxic they are. The next slide, notice that the horizontal axis is not 12%, but is 3%. And now you can see a very different picture, particularly for hydrochloric acid and for TA. They become very toxic. We see a decrease in mitochondrial activity. And so clearly, these are ones we might want to worry about. Another question is, how do different particle properties cause different damage? And again, different metals and different forms of these metals have different characteristics. And we know a lot about the chemistry of these nanoparticles. So here's a very dramatic slide that shows you we have 5 different nanoparticles. They're all approximately the same. So again, a nanoparticle is a particle that's less than 100 nanometers. So this is a tiny fraction of a micron. And you can see now that we've divided these or the data has divided itself into different categories. At the very top, you can see cerium oxide and barium sulfate, and even at the highest concentrations, given as milligrams per cubic milliliter in the solution above the slices, you can see that there's no change in mitochondrial activity. In contrast, you can see that silver, and then particularly in red, copper, at even very low concentrations, kills the cells and stops mitochondrial activity. Just an interesting anecdote that tells you the high throughput value of PCLS. The two at the top, cerium oxide and barium sulfate, were actually selected by the European Union and assigned to BASF, the big chemical company in Germany. And again, the EU government said different companies have to evaluate the toxicity of different nanoparticles. And the two nanoparticles that were given to BASF were cerium oxide and barium sulfate, the two you see at the top. And I would say they spent $5 to $10 million and 5 years or so to evaluate these two. They were aerosol exposures for 6 months. They sacrificed the animals after 6 months or even 2 years, both mice and rats, both genders, and then did histopathology. So this was a huge, expensive and time-consuming experiment, which is still not completely analyzed. And yet we were able to kind of replicate the same conclusion that these are not very toxic materials in less than a year. So we could also use this to answer other critical questions. Particle size, does that matter? We know that it does. Some of you are familiar with an air pollution criteria for PM 2.5, particulate matter that's less than 2.5 microns. And we've learned over the last decades that the toxicity of urban air pollution or occupational exposures to particles depends very much on particle size. And to tell you what we found and what has been known as well for other materials is that as the particle size gets smaller, the same mass dose becomes more toxic. So let me just show you this. Here's some data with silver. Again, we're looking at mitochondrial activity, and we're increasing the mass on the horizontal axis. And you can see that the smaller particles, the ones that are 20 nanometers, are much more toxic than the larger ones. This is an interesting result. This one compares cerium oxide nanoparticles with cerium chloride. And so cerium chloride is not a particle, it's actually a solution of cerium ions. And so from this and other studies, PCLS helped us understand that cerium oxide particles are not very toxic until they start to dissolve. And so we were able to show by a series of experiments that particle dissolution is very important, because it's really not the nanoparticles themselves that are toxic, but it's the ions, the metal ions that come from them as they slowly dissolve, mainly in alveolar macrophages. I've mentioned earlier that from the supernatant of the PCLS well, from each well, you can aspirate a small amount of fluid, and that fluid represents cytokines and other proteins that have been secreted into the fluid above the slices. And this is a list of cytokines and proteins that we've measured in individual wells. And again, lots and lots of data. And I'll just show you a selection of 5 of them that we produced from these same experiments. So after a day, we aspirated the fluid, and then we analyzed it for all these cytokines. And you can see the concentration of the cytokines are in the vertical axis on the left, and on the horizontal direction, you can see 5 different cytokines, including IL-6 and MIP2 on the far right, which is a chemokine for bringing neutrophils into the lung. And so if you looked at the red and blue on either side, the red is just the control medium, no particles at all. The one on the right, the blue one, is actually micron-sized particles. So they're about 2 to 3.5 microns. So they're respirable. They're small enough to go down into the lung, but they're much, much larger than the 0.02 micron, the 20 nanometer ones. And you can see this yellow bar of the small nanoparticles is elevated in every condition, particularly the two on the right. You can see that in MCP-1 and in MIP2, the signal, the cytokine signal is much, much higher. So I want to mention another virtue, which I think has been alluded to, is one of the nice things about PCLS data is that it correlates with information obtained at other levels. And so we really see a progressive approach. There still is a place for very high throughput cell-based assays that are in vitro, maybe 1, 2, or 3 cells. And then we see a role for PCLS, and then we also see a role for selected in vivo experiments. And so let me just show you an example of that. We then looked at some of the same particles and asked some of the same questions in some mice. But now, instead of each well being an experiment and getting 70 or 80 slices from a single mouse, now each data point is a mouse. And so you can see that's another advantage of PCLS. It doesn't eliminate animals experimenting, but it does greatly reduce the number of mice or animals or human lungs that are needed. So we basically looked at some of the same materials. We dosed them by intratracheal instillation, where we instill a small amount of material into the trachea and into the airways. And then we did bronchoalveolar lavage a day later, and we measured some of the things that you show that we see at the bottom. So again, here's the same question. Does particle size affect their pulmonary effects in vivo? And the answer is yes. You can see both for ceriums, this is cerium oxide, and silver. At the bottom are larger micron-sized or bigger nanoparticles, 50 to 200 nanometers. And the blue line on the left is for the 40 nanometer ones. And you can see that the neutrophils, which is on the vertical side, these are the number of white cells that have been recruited into the lungs, are much, much higher than it is for the micron-sized particles or for the bigger nanoparticles. And the same thing is true for silver. Again, as we go from a factor of 10 from 20 nanometer and compare that to 200 nanometer, you can see it's only the small ones that show a big increase in neutrophils in the bronchoalveolar lavage. And you can also notice there's a difference between ceria and silver in terms of the vertical scale, and that'll be even clearer here. So now again, these are the same rats and bronchoalveolar mice, bronchoalveolar lavage, and here we compare in each slide the silver nanoparticles to the cerium oxide nanoparticles. And you can see on the one on the left, so here are how many neutrophils are recruited into the lung. It's relatively low for cerium, but it's much higher for silver. And on the other axis, you can see the same thing is true for lactic dehydrogenase, a protein that belongs in the cytoplasm of cells but leaks out when they're damaged. And you can see the extent of damage is much, much greater for silver than it is for cerium. Here's the same thing with a chemokine with KC, and again you can see there's a dramatic difference between as a function of particle size, but also as a function of the identity. Note that the vertical axis on the left for cerium oxide goes up to 200 concentration, but for the silver it goes up to 3,000. So this single slide shows not only the importance of particle size, but also the importance of the identity of the nanoparticle. So let me conclude this part by saying that even though nanoparticles were present in this complex mixture, non-nanomaterials have varying and often dominant toxicities. And this has been a theme in our lab for some time. We've been very interested in excipients for drug delivery to the lung. We worked with Pfizer and Lilly on inhaled insulin. And again, there was a question about if you inhale insulin particles, does that have any effect on the lung? And we asked the additional question, what about the excipients that are also present? And we found numerous examples where the biomolecule that we want to deliver to or through the lung was not toxic, but the excipients that were added for a variety of reasons were. And so we're sort of the experts on the pulmonary toxicity of excipients. A lot is known about excipients for pills that are ingested or drugs that are injected, but very little is known about the lung. So that's one of the things we're interested in. Here are other conclusions. Particle size is critical. I've shown you that. Next bullet, silver is more toxic than cerium oxide. And I like the last one because we believe that PCLS data generally correlates with in vivo data, not maybe the precise values, but basic principles about particle size or particle characterization. And so we see this as sort of a general application to looking at new drugs or new toxic chemicals used for other reasons, looking in a progression from simple assays to ex vivo PCLS to whole animals. I want to close just with making a few other comments that I think you've heard in part. One is that excipients are important. Another thing that I think is terribly important, and this has been emphasized repeatedly, is to look at cross-species differences. It's a huge value for the same chemical, the same nanoparticle, the same drug to be examined in mice and rats and non-human primates and humans. And so one can make, again, conclusions and see what are the differences and what are the similarities between these other species and humans. Another thing that was been emphasized, and I would expand on it, is that we have at our disposal multiple models of disease, and that can be important. The data that I showed you were all with normal mice, but we do have animal models in our department and in Boston for asthma, for fibrosis, for pulmonary arterial hypertension, for emphysema, and for infections of either bacteria or virus. And so one can not only make precision cut lung slices of normal lung, but also of diseased lung and see how they respond to drugs or other treatments. And a great advantage, and one of the things we're glad we're part of this consortium, is to have access to human lungs. And again, some of the human lungs are normal, but again, there's data coming out of our department in Rama's where they've looked at the response of asthmatic human lungs. Another point that I think has been evident is that even though this can be a high throughput assay, it's also a high content assay. And so we're always getting new requests for new parameters to be measured. And so recent experiments have looked at DNA damage and repair in lung slices, at apoptosis, and other important processes. And another new technology that can be used is confocal microscopy. We were interested in whether the nanoparticles were they ingested by macrophages. And so one can take these 200 to 400 micron slices and one can interrogate them with confocal microscopy. Finally, I think as Rama's suggested, this assay is very nimble, and so it can respond to new crises and new needs, such as COVID-19. So just an example there that we've been thinking about is one of the concerns about the pulmonary failure is that there's a cytokine storm. And so again, we have been measuring cytokine responses in lung slices in response to different treatments. We also have some ideas about drugs that might reduce cytokine storms, such as IL-6. And so one could do that with virus, or even before that, very quickly, one could use other agents known to induce cytokine release and see whether there are drugs that can be found to reduce the expression of cytokines that we think may be troublesome. So I see great potential for this assay when used in all the ways that we've described. And I really am delighted to be part of this discussion today. Thank you very much.
Thank you, Dr. Brain, for providing an excellent example of an important application for PCLS. We do have time for questions, so there are some questions coming in, so please enter them on the right. And I will start. Dr. Brain, you mentioned species differences. Are there striking differences between humans and non-human species in regards to toxicity in your experience?
Yes, I think my feeling is that often if you looked at quantitative differences, the amount of a particular molecule secreted, there may be differences. But we've been reassured that in general, the qualitative differences, for example, silver being more toxic than cerium oxide, that this seems to be true across species. So especially these big, broad questions, and we found that for a variety of occupational exposures, different forms of silica, alpha quartz compared to crystalline, compared to regular quartz, or to other occupational dusts, where we like some of these because there actually is human epidemiologic data. And again, qualitatively,
right,
we find good predictive value between slices and humans. But I think the differences are also critical, probably particularly for something like a virus.
Right. Interesting. Thank you. I have a question for Rama, and then I'll start to go to the question board. Rama, in your presentation, when you introduced the TTM, you talked about that substrate. Can you elaborate on that material? It kind of reminded me of PDMS, but I'm not sure if that's what it is.
It actually is a variant of PDMS. Spot on. It's a soft, it's
Okay.
a variant of the PDMS family member. It's
Okay.
a soft silicone rubber.
Gotcha. Interesting. Okay. One question from the audience. How stable are the slices over time in culture? For example, the structure, gene expression, cell types. You mentioned 40 different cell types. How stable are those?
Joe, should I take that one, or?
Yeah,
sure. Why don't
go ahead, Rama.
you make a comment
Go ahead,
as well?
Rama.
Yeah.
Please.
So we recently actually just had a paper. It's in press now in scientific reports, where we have shown that we've done a careful comparison. Over time, these were in mice slices, and we've shown you can keep them going for 2 weeks and even up to a month. And the measurements we did is histological changes, and it's all in measurements of structural changes. We also measured preservation of airway contractility, and that was also well preserved. And we have a recipe on what's in the medium and so on and so forth. So the specific answer is that us and others have studied individual cell types based on applications. And overall, I would say most of what, gene expression changes, contraction changes, and structural changes are preserved. I would like to say, though, that and these were achieved, and these were in mouse PCLSs, that recent study, and these were achieved using pretty standard recipe items for the media. So there's nothing, very special that was added for this preservation.
I would just add two things. One is we're very excited about results that are showing us the importance of different formulations of the fluid, both in the freezing and in the slices after they're thawed out and used, that we're getting up now to 2 and 3 weeks. The one caveat that I would add, and I think Rama hinted at that, is perhaps the reproducibility and the survival varies on what you are measuring. And so there may be some responses that are well preserved. There may be other cell types or other physiologic responses which are more vulnerable. So I think there may not be a single answer to how long does the slice last. We may need to ask, we probably do need to ask, what particular cell or what particular function are you concerned with?
Right, so it depends on the endpoint. Okay.
Yes.
Another person in the audience asked, exactly. How stable are these PCLSs for, or is it possible to do immunohistochemistry?
I have personally not done a careful evaluation. So once fixed, how long can I keep them like that, for measurements of activity? I have not done such a careful evaluation. But I do know of colleagues. Yeah. But that's really the straightforward, the thing is, I honestly am not sure.
Okay. And
most of our assays
go ahead, Joe.
Most of our assays have been short-term, but for example, we're very interested in fibrogenesis. And there, if you're really going to see changes in collagen produced, then you may need to have a week or two. So we're very excited about exploring some of the potential for slices that can last longer.
Right. And is it possible to measure mitochondrial activity in the slices, Rama?
Yes, that's something we can repeatedly do.
Okay.
There's also the question of how often do you change the media, and how do the contents of the media change with time?
Yep. Okay. So, do the slices require special culture conditions for keeping them viable? Is it possible to create slices from a highly fibrotic tissue? I guess two questions there.
So the first part we've answered already. The second part about fibrosis. So there's actually one of two things that can be done. So you can obtain slices from fibrotic tissues, and there are publications on that. Though again, the caveat there is sometimes the lung tissue quality is so poor that it's tough to infuse agarose and slice. But it has been done, and there are papers around it. The other emerging class of studies, as Joe had mentioned this earlier as well, is you can induce fibrosis in the healthy human lung slice. So there's work, for example, from Darcy Wagner's lab, where you add a mix of cytokine, a cocktail of cytokines, and you can see 5 days, 6 days later, you can see deposition of collagen. You can see a fibronectin deposition, and you can look at what's released. So you could use it in one of two different ways.
In fact, what I think is interesting is that one of the features of lungs with disease is that usually different parts of the lung are very different. There's heterogeneity if you look at the distribution of fibrosis or emphysema. And so within the same 80 slices, if you look at them and sort them, you can divide relatively normal pieces and compare them to abnormal pieces. So that's the heterogeneity is a problem, but it can also be an advantage in detecting some of the differences responsible for the differences that are observed.
Right, and I imagine that the induction model for fibrosis is important because the frequency of those donors is likely to be much less. And so that actually reminds me of with AnaBios, our human DRGs for sensory neurons. We get those from consenting donors, and the frequency of pain donors is less. So we have inflammation and chemotherapy-induced neuropathy models. It's a very similar idea there. So question for Dr. Brain, is the shape of smaller particles different from the larger ones? If this is the case, does that make a difference?
Yeah, I think you ask an interesting question, which is being asked and answered, and that's, does the shape matter? Yes, so I think there's something is known about the fluid mechanics of particle deposition. And so we talk not about a physical diameter, but we talk about an effective aerodynamic diameter. And so that depends on the density of the particle and the shape of the particle. And so, a particle of uranium, for example, which has a density of 18 times more than water, even a very smaller particle, because of its higher density, behaves as if it were a larger particle. And so yes, I think shape is important, particularly in crystalline silica, which is very brittle and has sharp edges. It's different than amorphous silica, which is smooth. So I think, yes, the answer is shape matters, but it has not been studied adequately, particularly its relationship to toxic effects.
Right. Okay, so more work to be done. You both mentioned COVID-19 research and applications. Here's a question. Do you need to normalize cytokine levels to the amount of tissue per well?
Rama, you want to talk? You want to respond to that?
Yeah. So in general, what we do from our in our different measurements is we collect the supernatant. You can measure cytokine levels, but we do normalize it to total protein within each PCLS. We have we can measure it and normalize it in that fashion. But the other thing I do want to note is,
yeah, there is some variability in the mass of each slice. So again, that's a question that we all struggle with. How do we normalize them to take into account the variability and how much lung tissue is there? Again, there may be differences between slices that are dominated by large airways and ones that are dominated by lung parenchyma. So again, looking carefully, even in a normal animal, at differences, we keep track of where they come from, the top or the bottom of the lung, and so on. And so sometimes you want to have just pure parenchyma. Sometimes you want to include the pleura. Sometimes you want to emphasize large or small airways. That all makes a difference in what you find.
Exactly, and I just want to add one other thing, that if it's possible, we often will try and get adjacent, phases of a cut slice in the two groups, so you can study them. You have a similar starting point. So you get lung slices, from a certain core, place one slice, group one, the next one group two, and so on and so forth. So this way you start with an approximately similar starting point.
All right. Well, we are at the top of the hour. Dr. Krishnan, Dr. Brain, thank you so much on behalf of Dr. Getty and myself and all of AnaBios and the audience. Thank you for providing these excellent presentations. Thank you all for joining, and stay tuned for May, June, and July, where AnaBios will be presenting a series on pain presentations. So please inform your colleagues doing pain research to be on the lookout for more information. So again, thank you all for joining us, and have a great day or evening.